In this video, we are going to look about an important technique in molecular biology, which is Western blotting. So, what is this Western blotting? It is a detection of a specific protein using blotting techniques. So, we detect protein in this technique. It is also called as immunoblotting, as we use antibodies as probes. So, here, there we use in southern blotting or northern blotting, we use DNA as probes, but here we use antibodies as probes. So, it is also called as immunoblotting. So, what are the important steps in this? Techniques are protein separation, which is we use SDS page. Second is electroblotting. Third is anti-binding, so which we use specific antibodies to that specific protein target. And fourth is protein detection. So let's check all these important steps in detail. The first step is protein separation. So in protein separation, we use a SDS page technique. Already I have made a detailed video on that. Anyway, I will share the glimpse, important gist of this technique. So proteins, we impart a negative charge to all the proteins in the sample using SDS, sodium diodecyl sulfate. So it is an anionic detergent. So it will give negative charge to the proteins, and it will be as it has negative charge, when we apply electric current, it will move towards the positive electrode. So it will, when it move towards the anode, it will be separated. So the smaller molecules will be separated, it will be moving faster, because in the gel there will be pores, but smaller molecules will move inside the pores easily, and comes to the bottom of the gel towards the positive electrode very fast. But larger molecules will be moving very slow, because it will be hindered by the gel, the matrix formation of the acrylamide and bisacrylamide. So thus larger molecules will be moving slower, but smaller molecules will move faster, and it will be the protein molecules will be separated based on their size. So this is the first step, which is protein separation. Second step is electroblotting. So we give high electric voltage to transfer the protein molecules from the gel to the membrane. So the electric voltage is applied for transfer of protein. The proteins move towards the positive electrode. So here also we just move this protein molecules towards the positive electrode. The proteins are imprinted on the nitrocellulose membrane, the foam pad. So you can see in this picture, there's a blue color pad on the both side. It will be, and the paper helps to hold the gel and the membrane together. So it will be like pressing like a sandwich. The buffer helps to transfer the protein. So there will be a buffer will be flowing through this setup. So it will be transferring the protein from the gel to the membrane. So this is the electroblotting technique. Third step is blocking and antibody binding. So the blot is incubated with a dry milk, a blocking agent or BSA, to block the other sticky places, because we want our antibody to specifically bind to our target protein only. It should not be binding to other protein molecules. So we put a blocking agent so that it will be blocking other sticky places. First thing is primary antibodies is applied. So it will be specific to our protein of target, and it will bind to it. So we call it as AB1. Second is unbound antibodies are washed away. So again we wash it so that unbound antibodies will be washed away. Only our specific and primary antibody will be bound to the target protein. Third is secondary antibody, which are specific to the primary antibody is applied. So the second antibody will attach to the FC region of the primary antibody. So it will be like this. So this is our target protein. So primary antibody will be attaching to this, and the secondary antibody will be attaching over the primary antibody. The secondary antibody contains radio labeled isotopes or enzymes, which can used for detection. So for detection, the second antibody will be containing a radio labeled isotope or enzyme. Okay, so this is blocking and antibody binding. Protein detection, it is same like in other blotting technique also. So the radio labeled probes are attached and detected using radio autoradiography or other techniques like chemiluminescence. So as if it is having an enzyme, we will apply some substrate. The enzyme and substrate will react, and it will emit light. So when the light is emitted, we can find whether our target protein is there in the sample. If there is no any light emissions, means our target DNA is not present, or the antibody which we used for binding, it didn't bind with our target protein. So with this, we can identify whether our sample contains our target protein. Coming to the applications of Western blottings are, it is used as a confirmatory test for HIV and HBV, and it is used as a definitive test for bovine spongiform encephalopathy. Actually, it is simply we can say mastitis, mad cow disease, and a diagnosis of various other metabolic diseases. Coming to the Eastern blotting, it is same as like Western blotting only, but there are some small changes. Like here, there in Western blotting, we will be detect protein, but here we will be detecting post-translational modifications like methylation or distillation or acetylation. And the same Western blotting techniques will be used, like same polyacrylate gel electrophoresis, same electroblotting technique, same blocking procedure. But here there is a simple, small differences there. Here we use antibody binding technique, which is specific to the PTM, that is post translational modification, like which target the site of methylation or acetylation or lipids like that, and same detection technique. So there is no much of difference. Only the antibody which we use here will differ based on the post translational modification. So that's all. So at last in this video we discussed Western blotting and Northern blotting, its principle, its technique, its applications, and how it is done. So that's all.